So in this video, we will be talking about Down syndrome in detail. So Down syndrome is probably the most common syndrome that a pediatrician, genetic syndrome a pediatrician will encounter in his or her OPD. Down syndrome is an extensively described topic. It's impossible to cover in one video. So in this particular video, I will cover only the genetic aspect of Down syndrome. Down syndrome was first described by John Langdon Down in the year 1866. It's the first chromosomal abnormality described in humans. And the genetic basis, however, was described almost 90 years later in the year 1959 by Professor Jerome Liggin and Turpin. Once the chromosomal abnormality was described, 7 years later, we started to screen for Down syndrome by prenatal techniques. The incidence is around 1 in 792, and it should be remembered that even though we always attribute Down syndrome to trisomy 21, it's not the duplication of the entire 21 chromosome that causes Down syndrome. There are certain critical region in 21st chromosome, duplication of which will cause Down syndrome. So those critical that critical region is called 21q22.1 to q22.3. We know that 21st chromosome is an acrocentric chromosome, so there is no P arm, there is only Q arm. In the Q arm, there is this particular region where the Down syndrome related genes are present. The main ones are DSCR1, DYRK1IA, and APP. Why it is important is that sometimes we see a kid with Down syndrome and the karyotype comes out to be normal. Then we have to think about a segmental duplication of this particular region in one of the chromosomes. So there will be only 21 chromosomes. However, in one of the 21 number chromosome, there will be a small segmental duplication of this particular region, because of which there is manifestations of Down syndrome. That can be identified easily by a chromosomal microarray. Then I always had this doubt: why do we see trisomy 21, trisomy 13, trisomy 18, but not trisomy 1, not trisomy 2, not trisomy 3? The answer is very simple. 21 is a very small chromosome. Even 18 is a small chromosome. 13 is slightly bigger chromosome. However, it is not a very gene-rich chromosome. Gene-rich means there are chromosomes in which there are hundreds and thousands of genes. There are chromosomes, the number of chromosomes may not be as high as the other chromosome. And if you know the basics of cytogenetics, you know that chromosomes are arranged from 1 to 23 basis of their length. So 1 is the longest chromosome, and 21, 22 are the smallest one in terms of length. So 21 is a small chromosome. So even if all the genes in the chromosome are duplicated, still the child can survive. However, there are 10 times more genes in chromosome 1. So since the duplications of that many genes happen, most of the time the embryo won't survive beyond first trimester. So that's why we see certain trisomies more often, and most of the other trisomies, they get spontaneously aborted in the first trimester of pregnancy itself. So it's all about how many genes are duplicated in the trisomy. So I will describe the risk factors now. The single most important determinant is maternal age. We all know the significance of maternal age. Once the age of mother crosses 35, the risk is significantly high. Maternal non-distinction are more common than paternal non-distinction in attributing to Down syndrome, and 75% of the Down syndrome cases caused due to non-distinction happen in meiosis 1. This is one of the most important table. So if you do the karyotype of all the Down syndrome cases. In 95% of the cases, we see trisomy of 21, plain and simple. However, in 4%, we see translocation events. What is translocation? I will explain in detail. And 1% you can see mosaicism. So mosaicism means what? There will be some cell lines with 46XX or 46XY, which is a normal karyotype, and there are some cell lines with 47 XY with an extra 21st chromosome. Some studies say that because not all cells don't have extra 21st chromosome, the phenotype will be milder as compared to trisomy 21 without mosaicism. However, that is not always true because we cannot predict mosaicism of all the tissues just by testing the blood. For example, in blood cells, there can be mosaicism. For example, some of the blood cells will be having 46 cells will be having 47 chromosomes. However, that may not be case in brain. Brain will be having a different percentage of mosaicism. That may not be the case in kidney. That may not be the case in liver. So just by measuring the mosaicism or doing a karyotype in cell, even if we get mosaicism, we cannot always predict the phenotype. Rarely we can see isochromosomes or ring chromosome causing partial trisomy 21. So coming to the translocation part. So we know there are acrocentric chromosomes. Acrocentric chromosomes means they have a Q arm, and the P arm is extremely short. It is only, there are only few satellite repeat sequences. So it is known as acrocentric chromosomes. So there are two groups of acrocentric chromosomes, the group D and group G. D group consists of chromosome 13, 14, and 15. G group consists of 21 and 22. So there can be translocation events. That means I already told that these chromosomes have only Q arms. So two Q arms can fuse at the centromere, and this event is known as a translocation event. This can happen in a normal individual also. For example, me or you may be having a karyotype in which our for example 14 and 21st chromosomes are fused at centromere. Since there is no significant DNA loss, the carriers will be absolutely normal. However, when at the time of segregation, if a translocated chromosome goes into the fetus, there is a chance of aneuploidy in the fetus. So that's important. So sometimes what we see only 46 chromosomes. However, the child will be having Down syndrome. How is that possible? There will be two 21 chromosomes. However, a third 21 chromosome will be attached to some other acrocentric chromosome. It can be a 14, it can be a 15, it can be a 13. Sometimes what happens, two chromosomes, two 21 chromosomes. And one 21 chromosome will be absolutely normal, just like a normal 21 chromosome. However, the second 21st chromosome, it will be having two Q arms of two 21st chromosome. So in effect, it's a trisomy. Translocation constitutes for around 4% of the cases. And the importance of finding out a translocation causing Down syndrome is that there is a recurrence risk of Down syndrome if one of the parent is a carrier for this Robertsonian translocation. So Robertsonian translocation is a term described to explain the translocation between two acrocentric chromosomes. So the take-home message is we have to do the karyotype of all the Down syndrome kids, because only then we can predict whether there is any recurrence risk. If we get a plain trisomy extra 21st chromosome, we don't have to do the karyotype of parents. If we get a translocation, we have to check the karyotype of parents. So this is a table which explains how with maternal age the Down syndrome risk increases. So at the age of 35, the Down syndrome risk is around 1 in 400. This is the age which we consider as advanced maternal age, 35 or more. The importance is that once the age of 35 is attained by the mother, we have to offer NIPT test, that is non-invasive prenatal testing for aneuploidies, even if the Down's dual marker or combined screening first trimester is normal. That is important. And it should be remembered that there is no association with paternal age, birth order, ancestry, pregnancy interval, or blood group of parents. Coming to risk of recurrence. So you saw a child with Down syndrome and the parents asked you, what is the risk of having another Down syndrome child if they are planning for a pregnancy? Then if it is trisomy 21, then the risk is just 1%. So what you can advise the parents, the risk is small. In the next pregnancy, you can get an NIPT done or consult a geneticist and get the testing done, and the recurrence risk is very low. However, the catchy part comes in translocation. If the cause of Down syndrome in a kid is translocation, we have to do the karyotype of parents. Most of the time, the parents may not be a balanced translocation carriers. However, in some cases, parents will be a carrier of translocation. They won't be having an extra 21st chromosome. However, their 21st chromosome will be fused with some other acrocentric chromosome. So if there are two sets of, in fact, three sets of translocation. One is D group with 21. D group contains 13, 14, and 15. If father is a carrier, roughly the recurrence risk is 5%. If mother is a carrier, the recurrence risk is around 15%. Then translocation between two G group chromosomes, that is 22 and 21. And if a father is a carrier, then 2%, mother is a carrier, then the risk is 10%. The most important one is 21 to 21 translocation. So that means there is 100% risk that next child will be affected with Down syndrome. So translocation 21-21, you should remember that if you get a Down syndrome child in whom you see a 21 to 21 translocation causing Down syndrome, you should not counsel then and there itself that the next pregnancy also there will be 100% chance. You should do the karyotype of parents. If one of the parents is a carrier for 21 to 21 translocation, then the risk is 100%. Because in Down syndrome kids, if there is 21 to 21 translocation, 90% of the time it will be a de novo event. De novo event means the child has an event, however, the parents are not a carrier. So only in 10% of the cases, the parents will also be a carrier. So if parents are a carrier for 21 to 21 translocation, then the chance of getting another affected child is 100%, which is very important. And there is no evidence for increased prevalence of Down syndrome in second or third degree relatives. We can counsel the family if their second or third degree relatives have any apprehensions regarding that. Coming to diagnosis, always go for a karyotype. Only karyotype can detect a translocation event. So for rapid aneuploidy screening, we can do FISH or we can do QF-PCR. Usually karyotype, we take blood in heparin vacuotinal. Heparin vacuotinal is the vacuotinal with a green lid. We can do karyotype from amniotic fluid or coroniculum sampling. The turnaround time is important. For example, if a child is born and you don't need an urgent report just for the management, you can do a normal karyotype. The usual turnaround time is 10 to 14 days, and amniotic fluid and CVS will take around 15 days. If you want a rapid diagnosis, where do you want a rapid diagnosis? For example, the dual screen or combined screen or NIPT of the mother comes out to be positive for Down syndrome. Then you need a rapid diagnosis. At that time, you can use FISH in amniotic fluid or CVS. You do interface FISH because make a metaphase, it will take time. So in interface FISH, we can put the probes of 21 and see how many signals are there. And we can do FISH in blood also if we get a cord blood for testing. Remember that FISH cannot detect translocation. Even QF-PCR cannot detect translocation. Where do we use QF-PCR? the advantage of QF-PCR is that in karyotype we need live cells because we need to make metaphases. For making metaphase, we require cell division. So we cannot use cells which are dead. However, suppose a suspected Down syndrome child landed in your casualty. The child was very sick, had some cardiac disease, and the child succumbed to death. And everything happened in a span of 1 or 2 hours. Since the child is dead, if you take the blood sample and you wait for another 12 hours to, take the blood from casualty to lab, that sample may not be a good sample for karyotype. It has to be freshly taken sample, ideally. In QF-PCR, you don't need live cells, so you can so even if the child succumbed to death, you can take a EDTA sample. In QF, we use EDTA sample and keep it in fridge, and next day you can send it to lab. Even in QF-PCR, you cannot detect translocations. So best investigation is always karyotype. Fission QF-PCR, rapid, however, may not pick up translocation events. So that's it.